Good evening, Ishika. How are you doing? Okay. So today I'll be we'll be commencing the translation process. Okay. The production of proteins from mRNA. So in the last class or in the previous classes, we have discussed extensively about the transcription process where we learned about the processes that govern the formation of messenger RNA by using the antisense DNA strand of the double-stranded DNA as a template, right? And now those mRNAs would be exported out of the nucleus, right, through the exporting proteins, but those are not in your course RAN-GTP exporting pathway. But once they are exported, and post-translation modification has already been done in the nucleus itself. Now their half-life would be increased, polyethylening, 5-capping, and then they will be binding to the ribosomes. Now, how does the translation process pan out? Okay, so before jumping onto that, let me talk briefly about the ribosomes, right? The ribosomes in both prokaryotes and eukaryotes we will be considering here. So the bacterial ribosomes, if we put the heading ribosomes, ribosome in bacteria. So we have 70s ribosome in case of bacteria in the prokaryotes, right? And the mass is almost 2.5 million Dalton. That is the mass of the ribosome. And the two subunits that it can be divided into is 50s and 30s, right? Now 50s subunit, it contains 23s rRNA and 5s rRNA. 23s rRNA and 5s rRNA. And it is also made up of 31 proteins or R proteins because of ribosomal associated proteins, okay, plus 31 R proteins. Now 30s1 is made up of 16s, it is very important, as I'll tell you how, and also made up of 21 plus 21 proteins or R proteins, simply you can call it proteins only. So this is about the bacterial ribosome, you can say bacterial slash prokaryotic ribosome. Now when we come to the eukaryotic ribosome, what we have here is the mammalian ribosome. If we talk about it, is 80s. S is Swedenborg's constant or sedimentation coefficient, means how heavy it is, means 80s is heavier, so it would centrifuge, and during the centrifugation it would sediment quicker than the 70s, right? You might be knowing via laws of physics that centrifugal forces mv square upon r, more mass, you can say quicker time for settling down. So 80s mass is more. How much more? The mass of bacterial was 2.5. This one is 4.2 million Dalton. 4.2 million Dalton. Okay. And 80s is subdivided into 60 plus 40. 60s plus 40s. And 60 is divided into 28 s rRNA, 5.8 s rRNA, and 5 s rRNA. 28 s, then 5.8 s, and 5 s rRNA. Plus, it has got 49 R proteins. This 40s is made up of 18 s rRNA plus it is made up of 33 R proteins. 33 R proteins. Now you can easily differentiate between an eukaryotic ribosome and a prokaryotic ribosome. 60 plus 40, there 50 plus 30. Here sedimentation coefficient is 80, there it is 70, right? And there it has 23 plus 5s. This 5s is coded by the RNA polymerase. You can say this 5s, not that one. The eukaryotic 5s is coded by RNA polymerase 3, which also codes for tRNA, whereas this has only one RNA polymerase in case of prokaryotes, right? And then it has also got an extra 5.8 s rRNA, which is missing in case of the prokaryotic counterpart. There is no 5.8 s. The first one is 23, it is 28, it is 16s. Here it is. From 30s subunit, you have got 16s rRNA plus protein, you have 18s plus protein. Okay. Now, there is a term known as ribozyme. Okay. Why I deduced it like this is, there is a term known as ribozyme. So you might have heard that all the enzymes are proteinaceous in nature. Okay, but all proteins are not enzymes. This is an old saying. But the thing is, enzymes are protein in nature. That is all correct. Okay, but it's not mandatory that every enzyme will be proteinaceous in nature or will be protein biochemically. Okay, it's not mandatory that every enzyme would be biochemically a protein. They could also be an rRNA. So enzyme could be biochemically a nucleic acid also. Which type of nucleic acid? Ribosomal RNA or rRNA. The thing is, these 16 sRNA and all, mostly you can say in case of prokaryotes, 23 srRNA. Okay, this one. The 23 srRNA has got, due to its conformational property, its structure, its 3D structure, it has got the property of peptidyl transferase reaction. Means it can catalyze a biochemical reaction which is reminiscent of any enzyme, you can say, because enzymes also catalyze reactions, they are biocatalysts. This RNA, specifically this 23s RNA in case of prokaryotes, it does catalyze the peptidyl transferase reaction. Okay, and when it catalyzes as a biocatalyst, the peptidyl transferase reaction means the formation of a peptide bond between two amino acids in the formation of protein. You know that proteins are made up of amino acids. Now the bond between two amino acids is known as the peptide bond, and that peptide bond formation or that peptide bond synthesis is catalyzed by the 23s rRNA. Therefore, The ribosome is also known as a ribozyme because its rRNA can act as an enzyme. And ribosomes are also known by the name ribonucleoprotein because you can see here they are made up of rRNA and protein, both the things. So the thing is, this ribosomes, along with the property of being ribozyme, they are also known as RNP, ribonucleoprotein, because they are made up of rRNA and protein. That is why they are known by the name as ribonucleoprotein, rRNA and protein. So ribosome is also known as ribozyme. It is also known as ribonucleoprotein. So that's the introduction about the ribosome. Okay, it does contain protein. It does contain various kinds of rRNA. Okay, now any event I told you, whether it's translation or transcription or replication, in case of DNA, has three major steps. One is initiation, one is elongation, one is, you can say, termination. So we encountered initiation event in case of the mRNA, transcription event where we saw closed complex, then open complex, then TF2H providing the helicase activity in the open complex, and then the ternary complex where abortive initiation was overcome, promoter escape was overcome, and then the elongation, which was facilitated by GRI-A and GRI-B in case of prokaryotes and TF2S in case of the eukaryotes, and then termination. Termination was seen or observed by the action of row independent intrinsic or row dependent extrinsic factor. In case of prokaryote, in case of eukaryote, only dephosphorylation of the, you can say, carboxy terminal end or domain of the RNA polymerase 2. So here what happens is the initiation event would occur how? The initiation would happen at the ribosome. So once the, you can say, mRNA binds to the ribosome, its cognate tRNA would come and bind to the ribosome as per the codon present in the mRNA. But the thing is, what is the specificity or the structure of the ribosome? How many sites are present? How does the tRNA enter, okay, in the ribosome? So those things must be tackled first. So before going into that detail, okay, I would explain to you the various sites present in ribosome. Okay, the various sites present in ribosome. Now you have three sites: the A site, the P site, and the, you can say, E site as well. Okay, A site, P site, and E site as well. Now what happens is, I'll draw the figure. You have something known as, in the 16S RNA, there is something known as ribosome binding site known as Sine-Delgado sequence. Very, very important. Okay, of the ribosome. The ribosome binding site is present, not the Sine-Delgado sequence there. It is known as, it can be the anti-Sine-Delgado sequence also. Okay, so this is a short sequence of bases. Okay, that is positioned upstream from the coding region, is complementary to a portion of the 16S rRNA. Okay, so technically this contains the anti-Sine-Delgado sequence. The Sine-Delgado sequence is present in the mRNA itself. Okay, So what happens is recognition of mRNA occurs when a small subunit binds to form an initiation complex at the ribosome binding site. Okay, so the mRNA, okay, this is the 5 prime to 3 prime end. Here it may have the HD sequence, and at the 3 prime end of the 16SR RNA. Okay, the structure wouldn't be like this. It would have a tertiary structure. There would be a specific sequence complementary to this, known as the anti-HD sequence. Now, when they bind via complementary base pairing, does this mRNA bind to the smaller subunit, the 30S subunit of the ribosome? So, one more thing here, one mark question. This belongs to which subunit? 30S subunit of the ribosome. Okay, and the mRNA first binds to the 30S sequence of ribosome only, and after this there are certain factors known as initiation factors in prokaryotes and eukaryotes. Both we will be talking about the prokaryotes first, and then the initiation factors would come in and bind. Okay, and then what would happen is, what you will be having is the initiation event going on. Okay, then the initiation would be going on. Now prokaryotes, they use three initiation factors. Okay, three initiation factors are the main initiation factors which are used in the prokaryotic system. So means the bacteria, the model organism is the gram-negative bacteria, Escherichia coli. They use three initiation factors. They are the proteins, right? Initiation factors are the proteins, also known by the name as IFs. Okay, initiation factors. Now they are IF, you can say 1, IF2, then IF3. Okay, so before telling you about IF1, IF2, IF3, okay, I'll draw three sites. Okay, these are the structural, you can say, dimensions or the details of these three sites. They look approximately like this when an X-ray crystallography was done. Okay, this is the A site, P site, E site. Here it will be binding to the mRNA. Okay, this is the 30S subunit of ribosome. Now what happens is, in the beginning, in the very inception, the 30S subunit is dissociated, remains dissociated from the 50S subunit. Okay. Later on during the elongation phase, does this subunit become complete? Now this 50S subunit would come in and would join with the 30S subunit. This is the 50S subunit. So that's why I'm talking about the bacteria here. Otherwise it would have been 60-40, right? 50S subunit, 50 plus 30, and then this becomes a complete ribosome. But during the initiation phase, okay, this is absent, you can say. This one is absent. Only the half sites are present, half A, half E, and half P sites are present of the ribosome. A site is known as amino acyl site, P site is the peptidyl transferase, E site is the exit site. So before explaining this, I'll explain IF1, IF2, IF3. But before explaining IF1, IF2, IF3, I explained to you this structure of the ribosome, the three sites present in one ribosome and the associated mRNA, because IF1, IF2, and IF3, they associate at different sites or near or at the vicinity of different sites. Okay, that is why this site's acquaintance was very, very essential. Now, what does these factors do? Now let us start with IF3. IF3 has multiple functions. What it does is, it is needed to stabilize the free 30S subunit. Okay, IF3, if you talk about, very, very intricate functions are there. It is needed to stabilize the free 30th subunit, because during the initiation phase, these are the initiation factors. The 30th subunit must not associate with the 50th. Okay, it should be stabilized, and other function is the same, and prevent the premature association of the 50s subunit. Okay, why this is necessary? Everything will be explained. Okay, just hold on. Now, it also enables the 30th subunits to bind to the initiation sites in mRNA. Means it will facilitate the binding of what? The signed Dalgano sequence present in mRNA with the anti-signed Dalgano sequence present in the 16S rRNA, which is a part of the 30th subunit. Right? It enables the 30th subunit to bind with the initiation sites in mRNA, means with the, you can say, signed Dalgano sequence initiation site of the mRNA. Then what else it can do? It also checks the accuracy of the recognition of the first tRNA that carries the amino acid. So a tRNA which is carrying the amino acid, okay, it is known as transfer RNA, that I'll explain to you, okay, also in the next lecture, what is tRNA, very essential. The tRNA, for now, just know that tRNA is another type of RNA known as transfer RNA. RNA polymerase 3 codes for the, you can say, production of transfer RNA. Okay. In case of eukaryotes, prokaryotes, only one RNA polymerase is there. So in case of prokaryotes, RNA polymerase only will code for transfer RNA sequence. Transfer RNA and ribosomal RNA, they are the non-coding RNAs. They do not code for any proteins. Okay. Now, these transfer RNAs, they have anticodon sequences, which are the complementary sequences to the codons present in the mRNA. Okay, so I had explained to you the genetic code. If you remember, suppose this is the mRNA. Okay, this is the mRNA. Suppose this contains AUG. Triplet coding I had explained to you. CAG. Now AUG will have the anticodon, okay, present in the tRNA. It's complementary, and AUG anticodon, okay, or AUG as a codon is responsible for accepting methionine. So the tRNA molecule would have its anticodon. It will base pair with this one, okay, complementary base pair, and that tRNA would be associated with the amino acid methionine. And when this association is proper, when the base pairing is proper, it is checked that it has been proper, it will transfer the methionine amino acid, okay, the first amino acid, to the peptidyl transferase site or the P site of the ribosome, and it will dissociate and go away. Now this tRNA which is charged or associated with an amino acid, it is known as a charged tRNA, or the reaction, this reaction is known as amino acylation, the association of amino acid with tRNA, so it is also known as amino acyl tRNA. Amino acyl tRNA means that tRNA has been associated with its cognate or specific amino acid. And when it is dissociated from the amino acid, when the amino acid has been transferred to the ribosome, then that tRNA is without the amino acid. Then it is known as deacylated tRNA, just opposite of amino acylated, or I can call it an uncharged tRNA. Okay, these are the various nomenclatures that you will be finding in your book: charged, uncharged, amino acyl, deacylated. What does it all mean? What do they mean? So that is the meaning That is the scientific meaning of these things. Okay, the specificities of tRNA. Okay, I'll explain to you in the next class, like how do this tRNA work, how does the charging or amino acylation happen, that entire process. Then type 1, type 2 enzymes, amino acyl tRNA synthetases, the variable arm in the tRNA, the flower leaf model, the L-shaped model. Those things I'll explain to you in the next class itself. Okay, so this is the tRNA that we see. Now, the third function of this IF3, it checks the accuracy of the recognition of the first amino acyl tRNA, meaning that this recognition only, this recognition, that the codon must bind with the anticodon properly. It should not be able to bind with any other anticodon. The base pair must be accurate. Then only this will be transferred. Otherwise, some other codon, anticodon is there. It may be carrying some other amino acid, and AUG is specific for methionine. Suppose it is transferring glycine. Okay, then you are having what? A missense mutation. So that you don't want. You don't want any kind of missense mutation, right? So therefore, this IF3 also checks the accuracy of recognition of the first amino acyl tRNA. Accuracy of recognition of the first AA tRNA, I'll call it amino acyl tRNA. Okay. Now coming on to the specificities of IF2. Now IF2 is responsible for binding the initiated tRNA. Initiated tRNA means the first tRNA that would come in, meaning that is specific for the first codon. Most probably it should be AUG, because from methionine the peptidyl or the amino acid starts for of any polypeptide chain. So the first tRNA that comes and recognizes the first three codons or three nucleotides in the mRNA is known as the initiated tRNA. Now IF2 binds the initiator tRNA. It binds the initiator tRNA and controls its entry into the ribosome. Okay. Now this IF2 is very, very specific. What do I mean by binds? Means it is very, very specific for initiated tRNA only. I'll explain the mechanism to you. The meaning of this sentence is it does not bind to any other charged tRNA. Okay, only charged initiated tRNA or charged amino acyl tRNA. Okay, it will bind to. Now you'll be asking question, any other tRNA, if it is charged, how will it differentiate? Okay, so that is, I'll explain to you how does it differentiate later on. Okay, but the meaning of this sentence is it only binds to the initiated tRNA, doesn't bind to any other tRNA. Okay, because what happens is the other tRNAs which are specific for the second codon, the second triplet codon onwards, they enter the A site. The initiated tRNA must enter the P site. So there must be a discerning mechanism, a specific demarcating mechanism. We should be able to demarcate, differentiate between initiated and other tRNAs. The thing is, just understand this thing. I drew three sites, right? Now, P site, A site and E site. First tRNA along with its amino acid would enter P site, next tRNA would enter A site. This is the mRNA here. Now, why is it absolutely indispensable for the first tRNA to enter the P site? Because if it entered A site, okay, where would the next tRNA enter? That is the question. And when it has entered P site, the next one has entered A site. What happens is this amino acid tRNA's amino acid, this does a nucleophilic attack, causes a nucleophilic attack on this one, and then what would happen is the peptide bond would be formed, okay, between this amino acid and this amino acid, and this tRNA, what would it do is this amino acid would get dissociated from the P end, okay, and next step will be formation of this peptide bond. Now this amino acid has switched over to here. Now this tRNA is deacylated, and after that what happens is the ribosome, it translocates, okay, because it is reading this codon. Now suppose there are three more codons here, three more codons here. It will jump three nucleotides, okay. When it does jump, what will happen in the next cycle? This tRNA, okay, which is deacylated, would reach the E site. Now the at the P site, this tRNA will be reaching because it has jumped. So this will reach the P site. Now it has got two amino acids. Okay. Now the A site is vacant. Now the elongatory tRNA, the other tRNA, would come and bind here, acylated. Okay. Now the reaction will continue. Okay, and the reaction continues, and if you had put the first in the P site, how would this reaction continue? Oh, sorry, if you had put this first tRNA in the P site, how will this nucleophilic attack happen? Okay, it could have never ever happened. So the thing is, this is why the first one is put in the P site. Every one, if you put in the A site only, this would have never happened because it would have collided, then nothing could have gone in. So the first one is put in the E site, and then next every tRNA is put in the, you can say, A site only. The first one is put in the P site, so that this first reaction can happen, which will act as a chain reaction. Then other ones will enter or will keep on happening when this enters the A site as well. Okay. So the thing is how not to let the initiator tRNA or the first tRNA to enter the A site, how to prohibit its entry onto or into the A site, and how to make it enter only into the P site. That is the question. That is the mechanism is governed by the IF2. Okay, that is the IF2's mechanism, and IF1 also, it also adds a little bit of drama here. It will add a little bit of more reinforcement, you can say. So IF1, it binds to the 30th subunit, okay, in the vicinity of the A site, and prevents the first amino acid tRNA from entering. Okay. Now IF1, what it does is it will bind here in the vicinity of A site, and it will prevent the, you can say, binds at the vicinity of A site and prevents the initiator tRNA from entering the A site. The A site. So it does one reinforcement thing, that it aids in the process of not letting the initiator tRNA enter the A site. IF2 will make sure that it enters in the P site, and IF3 also, I'll tell you in the next class, it has an auto-inhibitory domain, or it has an N-terminal domain, not auto-inhibitory. which will mask the E site so that it doesn't go and bind or reach the E site as well. Okay, this is why IF3 it binds somewhere here. Okay, so this is how, and more explanation will also be given about the structure of the initiated tRNA, and first of all, what is a tRNA molecule, what are its structure, okay, or how does it pan out, how does it get synthesized, what are its most, you can say, accepted structures, the clover leaf model, the L-shaped model, everything will be explained in the next class. So I hope you are able to comprehend the lecture. Okay, okay then. Ishika, do you have any problem? Any doubt? Any queries? Any queries, Ishika? Okay, good. Okay then. Thanks a lot. Mm, Stay safe and let's meet you in the next class, Ishika.